So what I've talked about this morning is how we geneticists approach common neurological diseases, Alzheimer's disease and Parkinson's disease in particular, and how genetic approaches allow us to do two things. Firstly, they allow us to get better at predicting who's at high risk for disease. And of course, that will be important for identifying people who we can follow through the prodrome of the disease and get better at understanding, the very earliest symptoms. Obviously, we want to get better at identifying the prodrome in the longer term because we want to make therapies, mechanistic therapies, early in the disease process. And the second thing is to talk about how we geneticists, when we identify genes, we hope we're identifying pathways to disease. We hope the genes will identify multiple genes and proteins which are part of the same biochemical pathway. Now, I talked about Alzheimer's disease, and in Alzheimer's disease, there are simple autosomal dominant families where we know the mutations are in the amyloid and the presenilin genes. We've known that for some time, and we know that presenilin is responsible for amyloid metabolism. So the autosomal dominant forms of the disease have really pointed towards amyloid production as very important in the disease process. Now, as we've looked at sporadic, the much more typical late onset form of the disease, we've just started to make findings now in that area in terms of genetic predispositions. 15 years ago, apolipoprotein E was found by Alan Roses, but until the last 3 or 4 years, no other progress had been made. In the last 3 or 4 years, we found a series of genes, clusterin, CR1, PCALM, BIN1 are just a few of the top hits that have come out, but there are others too. And we are now looking at the function of those genes. And it's very interesting because 3 processes are coming out of these genome-wide association hits. The first is lipid metabolism, cholesterol metabolism, which ApoE had pointed at many years ago. The second is endosomal processing, and we think that might be involved in amyloid metabolism. We think amyloid goes through the endosome, so that might be directly involved in amyloid. And the third is the complement cascade. So these 3 processes are coming up as important in late onset Alzheimer's disease. And many years ago, Pat McGeer had shown that amyloid could activate the complement cascade. And so we're beginning to see then with Alzheimer's disease that its genetic variability in how the brain responds to amyloid might be one of the predisposing factors to Alzheimer's disease. So there's real progress being made in finding new genes and some progress in trying to understand how they map onto the disease pathogenesis and how perhaps they fit with the older findings relating to amyloid and the presenilins. Now, in Parkinson's disease, we've found 10 or 12 Mendelian genes, and we're now making findings in the genome-wide association for risk genes too. The first gene that was found is synuclein, Bob Nussbaum had found synuclein mutations in Parkinson's disease. We had shown synuclein duplications and triplications could cause the disease. And one of the findings that's come out of the genome-wide association studies is that genetic variability in the amount that all of us make of synuclein influences our own personal risk of disease, and that's obviously ties the Mendelian form of the disease with the more common so-called sporadic form of the disease. The other major finding is some very beautiful work started in Drosophila and being carried out by Helen Plum-Favreau now at Queen Square, looking at the early onset recessive forms of the disease. And what she has shown with her collaborators is that all of the genes actually which are involved in the early onset recessive forms of the disease are involved in mitochondrial homeostasis. So we are really getting the idea that mitochondrial homeostasis, the clearance of damaged mitochondria, is key to at least early onset Parkinson's disease. So overall then, the power of genetics is really helping us to understand the Mendelian and now the non-Mendelian forms of the disease, and we're beginning to see how all these new findings map on pathways for Alzheimer's disease and for Parkinson's disease. And of course, our hope is that through that understanding, we'll get better at designing mechanistic therapies.